Sorry, I had stopped the recording. So one of the most fundamental questions that we have, for some reason it keeps going backwards. One of the most fundamental questions we have is how we just categorize microbes. And so in a breakout group, what I want you to do is think of as many ways that you know of that you could use to categorize or classify microorganisms, okay? And then I need you to come up as a group, come up with one pro, a good thing about that method, and a con for each of those methods, okay? And then what I want you to think about collectively is also, would that method that you've just described work for both prokaryotes and eukaryotes? Or would it work for all prokaryotes or just some, some of these sort of like specific issues, okay? So what I'm going to do is create 6 breakouts. So there should end up being about 4 or 5 of you in each room. And so I'd like you to think about these things. So basically we want to come up with a method or set of methods, and then for each of those methods, a pro and a con, and then whether it would work sort of uniformly across all of life or maybe be different. Are there any questions about what we're going to do in the breakouts? Oh, everybody on board? Okay, so we'll see how this goes. I'm just assigning random breakouts for this one. So although we have one more person coming in, so I'm going to let Adam in before we do this. All right, so I'm going to go ahead and create the breakout rooms. All right, and then let me move to, I just need to rearrange one. Let's see. Where is Kate? Oh. Let's see, it looks like Caden isn't here yet. Okay. All right, so we'll go ahead and open all rooms. So just go ahead and accept your room assignment and move on in there. Hi, Jonathan. We're doing a breakout session where we're addressing these questions about methods that you use to classify or categorize microorganisms. So I'm going to go ahead and put you into, these are just random breakout rooms. So I'm going to go ahead and put you into room 5.
Sweet. Thank you very much. Sorry I'm late. I had some Wi-Fi troubles.
No worries. Okay, you should be able to just go ahead and accept it now. All right. So Beatrice, I don't see Caden on the participant list.
Hello, can you hear me?
I'm having a little bit of a hard, you're very quiet. Can you speak again?
Okay, hello?
Yeah, I can hear you now, okay.
Okay. Yeah, she might be just running late.
Yeah.
I'll try checking in with her.
Okay, no worries, but I'll just keep you here. Obviously, you don't need to be in the breakouts with the other other students. Yeah. So, and then hopefully we can add her to the room.
Okay, sounds good. Thank you.
Okay. I realized that in the future, it won't be too hard to move around. Once I create the breakouts, I can move you before we open the rooms. So should work out okay. So let me go to the chat. Okay, I'll go ahead and turn the recording back on now that I think we've mostly come back. All right, so what I'd love for you guys to do is maybe have one member of each group share one of the methods you talked about and basically just describe the method and give a pro and a con, and then state what kinds of organisms it would work with. So does somebody want to be brave and be the first to go?
I would say genetic sequencing.
Okay, and since I can't see everybody, can you just say your name? And,
Sure, this is Victor.
Victor. Hi, Victor. Thanks for. Okay, so talk to me a little bit about genetic sequencing. What do you mean by that?
So in order to categorize, we just sequence each species that we find, and unless it's the same species, they're all generally going to be pretty different. And I couldn't really think we couldn't really come up with a con. I think it's a pretty good way of categorizing, and it would work for both prokaryotes and eukaryotes.
Okay. So maybe not a con, but can you think of any limitations on that method in terms of like if you wanted to do it a lot for lots of different samples or lots of different cultures or something like that?
If we would wanted to do it for like a lot of different samples, we wouldn't get complete sequences. We would use like metagenomics, so just take a quick and dirty sample of like everything in the soil. We wanted to, I guess, just find out what's in there.
Okay.
But I wouldn't. Yeah, I guess that would be a con. No complete sequences.
Okay. So one of the things I was thinking also is just cost, right? So doing genetic sequencing is a lot cheaper than it used to be when I first started doing it. I won't say how many years ago, but it's definitely more expensive to do that than it would be some of the other methods that may be shared. So that might be something to think about. And so would that method work for all organisms?
I would say generally.
Yeah. Yeah. I mean, hopefully we can get a genetic sequence of any organism, right? So, all right. Thanks, Victor. So what group are you in? Do you remember?
3, I believe.
Okay. All right. Does somebody from one of the other groups want to go?
I can go.
Okay. Who is this? And what group?
This is Sandra.
Okay.
And I think I was group 5, I believe.
Okay.
So my group and I said that, so we said that our method would probably be the cell structure. And the pros for that would be, it will help us categorize the microbes quicker and differentiate between the both. And then the con would be that sometimes the structure can be unclear, so it's hard for us to determine whether it's a prokaryote or eukaryote. We tried to figure out number 3. We were kind of stuck on that, because we said that prokaryotes have different structure than eukaryotes, but sometimes it's hard to determine it under the microscope since they can be small and we can't really see the inside. So we kind of went with no on number 3.
Okay, so can I ask what specific method would you use to look at the structure? And really what structures are you talking about?
So it would be, for example, we would do the gram staining that would help us see the color and the shape of the microbe, whether it's a rod-shaped microbe or the,
Okay.
yeah.
All right, so that's a really good structural assay. So that differentiates which organisms from what other kinds of organisms. Do you remember? Like what groups
So
of
whether it's
organisms
like
should we use the gram stain with, I guess is my question.
gram positive and gram negative bacterias, and I guess
Okay.
that would also help us like see the shape, not just the color.
Okay, so we can see some of these shapes like you can see down here, but it works for bacteria. So now thinking in that way, would it work for the other prokaryotic domain, archaea, and would it work for eukaryotes to do a gram stain? Do you have a sense?
So so we actually kind of did struggle on number 3.
Right.
But I think we went with no because sometimes it can be hard to see the details under the microscope.
Mhm.
So then it would make us kind of struggle with differentiating what kind of microbe it is.
Yeah, although if you had a eukaryote that you did a gram stain with it, it would probably stain a color, but it wouldn't, you're not supposed to gram stain eukaryotes, right? So it wouldn't be telling you anything meaningful. Although what's one of
Yeah.
the things that we know about eukaryotic cells compared to prokaryotic cells, like if you look through the microscope? And anybody can chime in on this one. I don't want to pick on you, Sandra, make you answer. But if we did a gram reaction, we accidentally got a eukaryote in there, what would be a way we could differentiate that from, say, a bacterial cell under the same microscopic field?
The lack of peptidoglycan layer?
Okay, so it wouldn't have peptidoglycans. So the gram reaction, that's why gram staining is not appropriate for eukaryotes. But is there, I guess I'm thinking of a simpler, more fundamental difference.
They're bigger.
They're much bigger, right? So if you did accidentally gram stain a big thing, or you stained a mixed community and you had some eukaryotes in there, even if the color of pink or purple wasn't what you were wanting it to be, right? You'd say, oh, wait, this is a eukaryote, right? So I think you can use that method, but really gram staining is best used with pure cultures of bacteria, right? And an archaean will stain, but the results are meaningless because archaea do not have peptidoglycan. They have pseudopeptidoglycan, which is a related structure, but it's different. Okay, good. All right, is there another group that wants to go?
Yeah, I'm Jocelyn and we were room 2.
Okay.
And we said that morphologically we could look at streaking a plate of bacteria or any sample we had.
Okay. And what would that show you or tell you?
Oh, so that would be we could characterize them by their shape on the plate.
Okay, so colony morphology?
Yes.
Okay. All right. Yep. And that works quite well for some well-characterized organisms, right?
Right.
So definitely there are very distinct morphologies that would work. Are there cons? That's maybe a pro, but is there a con that you can see for that method?
Maybe although it's kind of easy to see different morphologies, maybe harder to distinguish them because there are some that look kind of similar to one another.
Yep. And is there a more fundamental concern you might have in terms of using this method to identify organisms?
I would think that there would be also a lot of other organisms, not just prokaryotes, but maybe some fungi or some other things growing in it as well.
Okay. Or you might have the opposite as well. Does everything grow on a plate?
No, having limitations as to like what is being able to be cultured. Like so far we've only been able to culture like 1% of microorganisms outside of their environment.
Exactly. So that could be another limitation. So would this plate colony morphology work uniformly for prokaryotes and eukaryotes? What do you
To a
think?
certain extent, maybe, but not fully.
Okay. And remember, you have to use a specific kind of medium to grow stuff. So it definitely would be very limiting, I think, for eukaryotic cells in many cases. All right. Any other methods that any of the other groups thought of?
Dr. Dillon, we discussed about the source of energy method.
Okay.
And for pros, we said that it would like require, it's like they need more. It's not always there for them, but for like because it's not always there for them. That's a con for that. Can that work?
So what do you mean it's not always there? Like energy? They're
Yeah,
energetic?
exactly. For the requirement, the nutrients.
Okay.
Yeah, for like you.
So when we talk about energy and sort of classification, what's a term that we will often use? I know one of the other groups was discussing that. Can somebody come up with a different term we might use for a method?
Autotrophs, heterotrophs.
Right. And when we talk about autotrophy versus heterotrophy or whatever, what kind of classification are we performing? Does anybody have?
You mean like metabolism?
Yeah. So we're talking about a metabolic classification.
Mhm.
I'm sorry, who was the first speaker? Because I can't see you guys.
Hamza,
Hamza. Okay. And what group were you in?
group 4.
Okay. Yeah. So metabolic methods can be a really good way. But there are limitations in terms of interpreting that, right? And oftentimes you need to use culture medium for that, right? So some of the same limitations we talked about with group 2's plate method. Okay. Would the metabolic approach work for all 3 domains of life?
It works for prokaryotes and eukaryotes because eukaryotes plants can use like source of energy photosynthesis.
Well, everyone needs energy, but like, can you give an example of a metabolic test that you could run? Like maybe something you did in General Micro Lab.
Oh,
Anybody remember any of those?
Like using blood auger.
Like the sugar. Yeah.
Okay, so using different kinds of media like PR glucose or blood auger or a sim test or something like that. Do you think that those methods would be appropriate for using with eukaryotes? No. Yeah, I see heads shaking. So they definitely wouldn't be. So it would be something that would really only work for prokaryotes, which is fine, right? There's pros and cons to all of these. All right. Any other methods that any groups talked about?
So just to piggyback on what Hamza was saying about the metabolism, I think that another drawback of that, just thinking back to microbiology, when we're doing kind of like those, find out what your bacteria is, like, everybody in lab would be like, do you think this is a positive or a negative result? So it's like very subjective whether what you're seeing is what you're seeing or not.
Yeah, methodology matters, right? And so with all of these methods, you have to do them properly and correctly in order to get them to give you the answers that are meaningful, right? So that's a really good point, Cecilia. What are some things we do, or what's a major approach we use